Hi everyone, welcome back to Science on Trial and Error. I'm Kasia Kuźmicz-Kowalska. I'm a PhD student in developmental biology and the host of this podcast. I'm so happy to see that our community is growing. Thank you for all the comments, all the love, and please keep sharing the episodes with your friends and colleagues. Also, please subscribe to Science on Trial channels to not miss on any of the future episodes. And if you want to see more of the behind the scenes and from time to time catch a sneak peek of my lab work, follow my Instagram account, Science on Trial and Error. I'm very excited to introduce this week's guest. Ewelina Kamińska is a Polish scientist and science communicator. She's a PhD candidate in the field of epigenetics and chemical biology at Ludwig Maximilians Universität LMU in Munich. She is working in the group of Professor Thomas Carell, investigating active demethylation pathways. In parallel, she is also pursuing a master's degree in endocrinology at the University of South Wales. Prior to her PhD, she got her Bachelor's of Science degree in human genetics from University of Nottingham in the UK. Ewelina is a first author of several papers from her PhD work, but she's also doing a lot of freelance writing in Polish. She's been a scientific expert for Forbes Poland, and she wrote 2 ebooks aimed at general public. One is focused on cancer and the other one on pandemics. Yeah, she's definitely a powerhouse. Ewelina is very passionate about science communication and is fantastic in explaining difficult topics in a much simpler way. Her mission is to get people to trust scientists more. Check out her Instagram and blog to find out more. Look for Ewelina. It's been wonderful to chat with Ewelina because she's one of the people who inspired me to do this podcast. I love that we get to honestly talk about challenges of working or even overworking in a competitive environment and the other challenges of the PhD life. Even though we cover a lot of difficult topics in this interview, I think you will still end up feeling motivated and inspired as Ewelina shares how she actually manages to do it all and have a life. I'm not going to reveal anything more, just listen and enjoy. Please welcome Ewelina Kamińska. Hi Ewelina, thank you so much for being here today. I was really looking forward to this interview, and I'm glad we managed to find the time to finally record it.
Me too, seriously. It was so hard to find this date that we are both available, and sorry for keeping you waiting for so long, but I hope it's worth it.
No, absolutely no worries, and I'm very glad we managed to do it right now. Where are you currently?
In Munich. I'm currently working, finishing my PhD thesis, and slowly packing my stuff as well because I'm leaving not too far from now. Yeah, it's been a very busy time for me, but yeah, most of the time I'm spending in Munich at the moment.
I see. So do you have already a thesis defense date set up?
No, I don't have a set date yet because at the LMU we have such system that first of all we write the whole thesis, then we give it to the professor for the final checks. Mm-hmm. Then we send it off to our reviewers, and when they say they're ready, we set up a mutual date that we can all meet. So far, I think it's going to be an online defense, which is something new and something that is maybe a bit more difficult for PhDs at the moment. But let's see. I hope to be finished by the end of the year.
That's really nice. The schedule must be tight. As soon as you finish writing, then you are kind of waiting and you can wrap things up. But yeah, exactly. It must be a lot. Usually what I like to ask my guests at the beginning is whether they actually listen to music at work. The idea is that after a couple of episodes, I'm thinking of even having a small playlist. And I was wondering if you are this kind of person that listens to music in the lab. Are you listening to podcasts? Or you have to be really without any sounds in your ears to focus?
Well, it depends what I'm doing. So when I'm doing like brainless pipetting, really something that I don't need to focus on at all, or some kind of maintenance and cleaning stuff, then I listen to podcasts that, people are speaking and I can actually understand them. When I do something more like focused, something that requires from me a lot of more attention, then I would listen to some pure music without any lyrics, which is usually something alongside jazz and classical music, because then I feel like I'm focused and nobody disrupts me because they see I have my headphones on.
Yeah, I get it.
And I'm this kind of person in the lab who always wears headphones, even if I don't listen to anything. I just don't want to be disrupted when I'm doing so important
so
stuff.
well.
So yeah, this is the two types of basically activities. So I have different set lists, playlists for both.
Yeah, I kind of do the same. I mean, when I'm doing something very repetitive, for example, I'm at the Kreuzstadt, really mindlessly just moving my hands and doing the same stuff over and over, then the podcast is very easy to follow. Where I have to really focus, I'm doing dissections, then I go for music, or I just put the headphones to really be not disturbed.
Sure, and something that I really discovered back in high school, but rediscovered during my PhD is the earplugs. Seriously, at the times when I have to write a lot or I have to think, but I cannot be disrupted by any other noises, my productivity and work efficiency is rising a lot.
All right, so how about we start by talking about your current work, the PhD that you're finishing. I know that you're doing PhD in epigenetics, right? You mentioned LMU, which is in Munich. And yeah, I would be very curious to hear more. I had a look at your papers, but honestly, they're also so chemical. It's quite impressive. So I guess this is a bit of a biochemistry focused project. Can you elaborate maybe a bit more, then we go into details?
Yeah, sure. So basically, it's a chemical biology, not biochemistry, which is a bit different. It's a brand new branch of science. Not maybe brand new, but at least more novel than biochemistry. And we are basically using chemical tools to investigate biological processes. And what does it mean is I moved from totally biological background because I studied human genetics to do my PhD in a chemistry department, which is kind of, it's a big move. And I had to literally study Masters of Organic Chemistry during my first year. But at the same time, I think this is the knowledge everyone needs who is working in epigenetics field, because chemistry of nucleic acids is something in a very biological context, but it's a chemistry basically. So I really wanted to pursue my further career in a more chemical environment, because I knew if I want to purely understand the meaning of epigenetics, I also need to understand the chemical processes. Coming to the lab, of course, I'm still doing more biochemistry stuff in the lab, but I'm working alongside synthetic chemists. And this is a great experience to actually learn how all the nucleosides are made and how to synthesize DNA. Because when you want to work in, for example, medchem or any kind of drug development companies or research in future, you need to kind of have an idea of how to synthesize a drug.
Yeah.
And then you need to also have an idea how it would work in a body, in a cell, right? And I think a lot of chemists are synthesizing, seriously, sorry, chemists, but I think they're synthesizing compounds just for fun. And very often I met with situations when the chemists were synthesizing compounds. They are fluorescent, but they are fluorescent at such wavelengths that we have no microscope to see it. Yeah, it's something that is not soluble in water, but in loads of, DMSO and stuff. And this we cannot use in the cells either. So I think really having the knowledge of both gives us the advantage that we can, in future, create better basic research, more applicable basically in science.
Sure, definitely. And you mentioned you work with synthetic chemists. How does it work? Is it actually possible for you to reproduce certain modifications that are epigenetic modifications completely in vitro from scratch with the chemist? Let's say, how much to the beginning can you go? From how small building blocks can you actually build?
The possibilities are amazing. First of all, maybe I would say what my project is about. It's epigenetics, and it's to do with active demethylation, because we know, of course, we have methylation of cytosine in the DNA. For people who are not familiar with this is needed for the cells to be able to differentiate into different lineages. Different methylation patterns allow certain genes to be active and certain genes to be silenced. Because, of course, almost all of the cells in our body have exactly the same genome. And they don't need all those 21-something thousand genes at the same time. They just need specific genes to allow them to function properly. So people are investigating methylation patterns, but also there are some people like me who investigate demethylation. Basically, the methylation is never constant. It changes. And this is like a fresh topic in the epigenetic field. So we are not really sure why exactly it happens. Very often it happens in like cancer cells, for example, or when you have differentiation from stem cell to like somatic cell. And sometimes it happens by the error. Cytosine will get methylated by accident and then you have to revert it back. And sometimes you just need to switch on a gene that was switched off before. And in order to do this, the cell has very complicated mechanism, because if it's meant to be like a switch on and off, it would be maybe error prone.
Yeah, so it needs more checkpoints.
Exactly. So we have a whole cascade of reactions in order to achieve cytosine that is unmethylated again. So we start with methyl cytosine, and then there are PET enzymes, which are oxidizing this methyl group into hydroxymethyl cytosine, and then pharmacytosine and carboxycytosine. And what happens then, you have one pathway that is very well known already. So you have a TTG enzyme, which directs by fusion reverse. But I'm investigating more like a direct route, because a base excision repair, it's a process that is very energetically demanding from the cell. And also, during BER, you have a risk that you will have like double-stranded breaks in the DNA, and this is something you don't want. So we as scientists assume that there is another system that allows the cells to deformylate or decarboxylate in a way that it will not cause so many issues. We already published 2 papers on deformylation, and the most recent one, which came out last week, is about decarboxylation, because we think we found the mechanism for this.
These events of demethylation, actually, how often do they occur, do you know? Like you say, it happens in cancer cells, which would suggest that it's not maybe so frequent, but I guess it must be happening in normal somatic cells from time to time as well, right?
So basically, yes, it happens everywhere. We just find the very high levels of the deformylation and this demethylation in the somatic cells. And of course, in embryonic stem cells, you have this event of active demethylation where you have this epigenetic reprogramming.
Of course, yeah.
So when your mother's and father's epigenetic marks need to be removed in order for your own ones to appear. So it does happen, but of course it's not this kind of process that we can see like every day happening like crazy, because even the amount of Fc in the genome, so formal C, is super tiny, right?
So it's so transient that you cannot catch it. That's what you mean.
Exactly. It's really something that we can just assume and postulate, because what our lab does is finding the mechanism. But the actual biological relevance, this is for more biological labs to check. You asked before what kind of synthetic tools we can use in order to study this. Of course, if Fc changes into C, we are unable to see it without marking the Fc.
Yes.
And marking the C, so we know that it comes exactly from this formal C. So the guys in our lab are synthesizing a synthetic labeled nucleoside that we feed to the cells. By feeding it to the cells and getting it incorporated to the genome and using the mass spec, we are really able to see which C comes from this Fc that we fed to the cells. We also need isotopically labeled standards to be able to see it on the mass spec as well. So this is a lot of synthetic chemistry.
You said yourself you are working on cancer cells. Are you actually looking in different cell types?
Yes, so we went through loads of different cells because we tried to find the perfect system to study the formulation.
Is there a model that works really the best for you currently? Did you find it?
There was for the formulation, yes. It doesn't really work out with the carboxylation, but this is a different story. So the formulation was really a perfect system because, first of all, we noticed that some somatic cells that are more like neuronal, like neuroblastoma, for example, they deformulate quite a lot. So then we went further and we wanted to see different like neuronal progenitors and stuff. And then we came across those very special iPS cells, and they are called INGN. And they are basically neurogenin-inducible iPS cells that were just normal fibroblasts back in the day. Someone reprogrammed them back into the stem cell. And what we are doing is we are converting them in a new way to neurons. And I love this process because from those very like fibroblast-looking cells, you have, after a few days, fully grown neurons with very long axons, which are, from one side of the plate to another. And these cells, after the second day, they stopped dividing. And this was a perfect situation for us because we could study if the formulation happens without diluting our samples.
So these cells are good because they stop dividing, but also, I guess, they in general have higher frequency of these events. Is this what you're finding?
Exactly.
So this is kind of interesting from the functional point of view, because if you consider that neurons are the kind of cells that don't really regenerate or like they stop being produced. I may be going a bit out of line here, but yeah, perhaps this is a way for them to kind of be a bit more adaptable to whatever is happening. Is that something that you think?
We know that neurons have quite high methylation levels anyway, higher than usual, and higher hydroxymethylation levels as well. So we think it has something to do. I'm not a specialist. We have other people in the lab who are specialists in neurons, but we think it has something to do with adaptation, maybe memory formation, or something that is quite demanding. And of course, we know that plasticity of the brain is something that doesn't happen in any other tissue, right?
What about age of the cells? Mm-hmm. So is this something that becomes more frequent as the cell becomes older?
This I didn't study specifically because most of the cell lines that we have and we culture anyway have high passage numbers. We would have to really have, I think, more of a patient tissue sample. And this is more like translational science, which is something that really interests me for the future. But we are kind of limited to basic science in the lab that I'm in at the moment.
So coming back to a bit more basic question, are there any hotspots that you find? Whether it is, like CPG islands are very hotspot for methylation. Is this also happening more frequently there, or is it maybe related to some group of genes? Do you have this kind of knowledge already?
Maybe it's worth to show the difference between sequencing and mass spec, because sequencing allows you really to see a whole sequence of the genome from like point A to B. You know exactly what nucleotides are one after the other. But sequencing so far doesn't allow you to see such modifications of cytosine. The most we can do so far is methyl cytosine, right, in a bisulfite sequencing. But the rest is really something that is not that successful yet. And what mass spec gives you is the more like quantitative data. So basically, you can take your genome, you can chop it into nucleosides, and you can see what's there.
How much of the certain modification is in total? Okay.
Of course, you can chop your genome and you can just pick up the points that you want to check. For example, like you can enrich it for CPG islands and stuff, but it's still not like
as informative. Yeah.
So, yeah.
I mean, this all sounds very interesting, and I guess it must have been quite a big transition. As you mentioned, you had to learn the organic chemistry and you had to change your way of even thinking about nucleic acids in a completely different way. But as you mentioned, it's something that is currently a very hot topic and a very interesting area of research, especially it's a basic research, but it's very easily can be taken to the translational research or drug development. Is this something that you are planning to do next? Is this the direction where you want to go?
Oh, I hate those kind of questions because I have like tons of ideas for myself, especially at the moment when I'm like almost finishing one chapter and just, getting myself ready to start the next one. I never make it easy for me, for myself. And I get very easily bored with stuff. And then when I'm bored, I'm not so productive. That's why I'm always jumping into deep waters. I must say, I don't have like a set idea for myself for a far future.
Mm-hmm.
I have an idea for another like 10 to 12 months so far. It's a kind of a project that I cannot really talk about publicly yet. And it's something that is not necessarily connected to research because I decided to take a step back in order to really think through what I want to do in life. Because when you are so deeply in your basic research and you're like pivoting like crazy and you have multiple experiments that didn't work and you always try to find solutions for them, you never really have time to think what you want to do next.
Yeah.
In order to, to pursue your further career in science, you have to have some ideas. So far, I want to take a step back and I want to spend some time on just really thinking what I like doing, what I want to do. Because during your PhD, you just see one experiment and the next. And whether you like to do it or not, like you just do it, right?
Because
this is the next step. This is the next question you want to ask yourself.
But also you lose track of what is out there. I feel like you get so involved into your narrow field of working. You get involved in your project. You get more and more committed. Of course, you could see yourself taking the path that is kind of a very conventional path from wherever you are. Like for me, being in developmental biology, I could go into a different model organism, I can go into different question, but there's a certain path that I could take. And I was super certain I'm going to do it. And now I find myself coming closer to the end that I actually think, okay, maybe I want to do something translational, but I never did it. So how do I go about this? Of course, I know the obvious path, but like there's so many other things out there. And Science F is moving very fast forward and there's a lot of interesting initiatives happening. So I think that's a good plan.
Absolutely. The thing is, when I've been in this lab for so long and I've been working day and night and all the weekends and everything, I stopped having fun just by doing simple experiments. So I won't, miss it a bit maybe. Also, when I look at the, job offers, for example, and the whole list of experience you're supposed to have as a PhD student, it's kind of ridiculous. This is also wrong thinking. So only like a few weeks ago, I realized that anyone who comes to our lab as a postdoc is anyway like a new person.
Yeah, they have to learn.
You may you may use a different mass spec and you still don't know how to use it. You need to learn from scratch. I think I need a few months to work on my own like self-esteem, on myself, because apart from doing my PhD, I've been doing loads of other projects as well. I'm going to do something quite exciting and I hope you will find out about this like within, maybe next 6 months or so. And I think it's still something like very cool and very needed for the society. But maybe it's not something like a conventional route, like you mentioned.
No, but I think if these pandemics have given us anything good, it's actually the realization that a lot of things can be like shaken at their base and they can still survive. Science had to adapt, academia had to adapt, and work market had to adapt, and suddenly things that were frowned upon are new normality. And I feel like it's good because it's opening up many doors and it's making people a bit more open towards just non-conventional ideas and non-conventional paths. And yeah, good luck for your secret project.
Thanks.
Coming back to your gigantic to-do list, I feel like I've seen it on your stories at some point. You are actually doing also a master's studies currently, like in addition to your PhD?
This is something that I'm doing, and I still need 1 more year to graduate. And this is also one of the reasons why I don't want to start a very engaging new scientific project, because I just want to finish the ones that I started. Probably as a Polish person, you know very well that we have this society, like, I don't know, like this mentality that we need to do everything super fast, super early. We need to graduate before we are even born, I think. I realized that I've been trying to follow this path. I really wanted to, graduate as soon as possible. When you try to be so fast and efficient, first of all, very often you're too young to realize some things. Things just need time, especially science, time to sink in, to make us really, to be experienced people. This is something I realized not long time ago, really. You think when you're younger that when you finish all the steps that are waiting for you, that when you graduate, finally you will get this amazing job, will be well paid, and job of your dream, and this is it, and this is how you're going to comfortably live for the rest of your life, and then you can rest. And this is not true, because the world is becoming more difficult with those next steps, more competitive, and this is when your energy expenditure starts.
So you also had this, like a moment of disillusionment when you suddenly realized that it's not necessarily that your hard work is going to translate into a very successful career. Because this is something that a lot of my guests are mentioning, a bit of a dream thing situation that we come to academia, we work really hard, and we are spending a lot of time in the laboratory or doing our research, and then suddenly there's this moment during the PhD when you realize, okay, I'm doing this, but everybody else is also doing it. And also, it doesn't necessarily mean that those who work the longest hours, the hardest, or whatever you call it, will be the ones that will be the most successful, because a lot of things come into play, connections, like personality traits, and a lot of random things that you cannot really predict. And it's a bit of a soul-crushing moment, I must say. I agree with what you said about coming from Poland and feeling like we somehow have this aversion towards doing nothing as it's a bad thing. But another thing that I think is, I don't know if you will agree with me, is that we also have a problem with, let's call it believing in ourselves, in a sense that, I don't know if you had this issue when you went abroad, but for me it was something that was really hitting me hard. I was very afraid that somehow I will be worse than everybody else, and I always was very careful in all of my applications not to oversell myself, trying to be honest. But actually my supervisor from my master's, we were talking about my applications and she read my letters and she was like, this is all wrong, you have to rewrite it. You have to start believing in yourself, and you have to be kind of more bold in your statement.
Yeah, this is the story of my life.
I was just like, how do I do this? Because we are not taught this. We are not taught this boldness and this self-belief. And not even mentioning that as female, we are also kind of taught to not be braggy and not putting ourselves out there.
This is absolutely true. And I think it's like a systemic problem that starts from the very beginning, because first of all the parents want to bring the kids to school as early as possible, because people have this idea that if your child finishes school early, then has some advantage in life. And very often those kids, they may fall into so many issues. They may just feel wrong in the wrong place, being at school so early, that it's basically destroying their whole career. And also, we are coming to school and everyone tells us this is like work. We need to just do our homework and pass the exams with the best grades, which I think is also all wrong, because we all know that the exams are just not checking your knowledge at all.
No.
And I think this is the start, because in the US or in England, I noticed like kids are taught, you can be the next president. You can be anyone you want. You have time to develop.
Yeah.
For sure, this is one of the things that came after, our very difficult history. Maybe we were basically for many generations brought up in a very, very hard times. Because of this roughness, maybe now we think, okay, when we have chances, we should just go for it and then try. And the thing is, people then go to university just after they finish their high schools, to any kind of course, and they realize after 2 years that this is not it, and they need to change it. And of course, this is also a moment when they get judged by other people, because, how can you be so undecisive?
And giving up and, changing your mind.
And the more we read about other scientists and other brilliant people, we realized they were like school dropouts, or they actually stopped studying philosophy for 10 years and then came back to study like physics and they became like great physicists afterwards. So I think really because of this treatment, then afterwards in our adult life, we don't appreciate ourselves. And this is where you said our CVs don't look as good. Maybe that's why we don't get those jobs or those university positions, because we underestimate ourselves for sure. Mm-hmm. And we cannot really say that, oh, we know it all. We can learn. We are capable of doing all these things. I think in Poland it's very difficult to admit, whether it's male or female, I'm good enough. And I can do more, but what I already achieved in life, it's already okay. All of it I realized when I moved to England to finish my high school, when I was, doing my usual work, and then all people around me were saying, oh, you're so hardworking, Ewelina. Like, when do you rest? And I was like, well, I rest on holidays, weekends for doing my part-time. How long can you live like this? At some point, you realize that your health is maybe not so good anymore. You have some stomach issues, and manage your stress.
Anxiety and all these kinds of things hit you hard.
Exactly. And this is where it all happens. And basically, this is exactly what happens during my PhD. So I made this mistake twice. Of course, I couldn't say no to all other initiatives, whether online or offline. And then I realized, now I need to rest.
But it's good that you realized this. Honestly, to me, a wake-up call was when I actually got really, really sick. Also because I moved to Austria to study, and I was actually in a long-distance relationship. So my husband, he was doing a PhD in Poland. And I always felt like, okay, he's there, I'm here. The more I work, the faster I'll finish. Or,
Yeah, you have time for it because you don't need to see your husband.
Also, I moved here to do this, so I should really do it. I shouldn't waste my time. And of course I was tired, but I was like, okay, well, push through it. And we do like our job. It's not like we don't like it, because otherwise we wouldn't do a PhD. But then I get sick and I was like, okay, I have to really reevaluate how to do this. The biggest problem that I have, I think, even at this moment, I feel like now that I'm sick, I work even more efficiently with time and productivity than before, because I don't want to spend too much time to exert myself physically. But on the other hand, I still get into this mindset that somehow I'm being lazy, or somehow I'm not doing enough, or you wanted to do a PhD, you should be up for whatever comes your way. I get into this guilt, which is a very stupid thing. And it's very hard to keep yourself in check. I feel like talking to people helps, because you realize that everybody feels like this. But it's a very slippery slope, because you just get into this guilt set, and then, okay, I have to make up for it. And it's hard to take care of yourself, but take care of your project. And I feel like taking a step back from this kind of work may be good. Also for looking for a job.
Yes, absolutely. And, the environment that we're working in, this PhD environment, is absolutely tough. It's full of very ambitious people who are overworked as much as you, who have the same ambitions, kind of are weirdos, who tend to overwork themselves. And then we look at our neighbor and we see that she's working 2 hours longer than you, and you already feel guilty, right? And this is very important for future PhD students, to look for a lab where your boss is already so clever to not, force you to work long hours. Maybe back in the days, competitiveness helped. But nowadays, there is absolutely no single discovery or a brand new drug or system which is made without a super interdisciplinary team. And I know they are professors and they're group leaders who are now approaching it in a different way. So they have some experience in coaching. They have some experience in real leadership, not fear leadership. I really think the best thing would be for people who want to pursue their scientific career further into the PhDs to join a lab for a day or two. Just do like a, training day and see how the people operate. See if people like each other, see if people talk to each other, and see how the professor is treating people at the same time. Also, a PhD is like real job. You just need to do everything by yourself. There is no structured programs usually. And this is tough. And nobody is here to tell you, oh, it's really tough, I understand you. No, everyone is like, where are your results? For sure, I think people should really, especially now when it's not that difficult to get maybe it's difficult to get the PhD positions, but at least you have a choice.
Yes.
So it's better to go for maybe a less known professor, but someone who will treat you well. This is what I'm saying.
The funny thing is, anybody who had this kind of experience of having difficult work environment, let's call it like this, I feel like now when you make your next step, you're going to be much, much more aware of what you want to have from your next job. And getting into PhD, you still feel like, oh, I get so lucky that I get accepted, I should go there because they want me. And people just don't realize that, first of all, it's all right to have questions, and it's all right to just be a bit more inquisitive about like how the lab situation is. It's not something offensive. Anybody who's a good PI will understand that actually you want to fit in and you want to see if it's a good fit. And it's beneficial for everybody to check this and to make sure that it's true. I want to talk about your different initiatives, and I'm sure we're not going to be able to cover all of them. I'm sorry. I actually encountered you a while back when I saw your blog. I rediscovered you on Instagram again, where you actually do a lot of good work getting people acquainted with how scientist life looks like and how scientist work looks like, to kind of get the society to trust the scientists more, which I think is really an important, very important mission. I would like to start with that, and I would like to ask you first, where did this come from? Was it something that you always felt like you want to do? Was it something that was prompted by, I don't know, discussions in the family, something that happened? How did the idea for this get born?
For being a science communicator, right?
Yes.
Oh, this is it brings back really good memories, because actually from very early times of my life, when I just learned the letters, I always wanted to write. I was really, I think I was born to write, basically. And I had ambitions to become a journalist, but it never worked out. Yeah, I was stealing, other people's computers just to open like a notepad. And I was typing like kids' stories, very basic, additional chapters to Winnie the Pooh or something like this. And since then, I was always into writing, but the idea about the scientific blog or popular science blog, so I really wanted to do it, but I always felt inexperienced enough, of course. I always thought I don't know enough to be able to write about science. And then I realized, with the help of course, people around me, that I probably know more science than like an average human being. Even being at the university, I started to write about very like basic things. I started in English, but then I found a niche to really produce something in Polish. And of course, when I started Instagram, it took me a long time to get into Instagram. And of course, it was at the beginning in English as well. And then I switched into Polish and I lost all my followers and my motivation went down. And then I rebuilt it from scratch, basically. So this was something that was in my mind for a very long time. And I think I have this additional skill for explaining something very easily. At least people say that they understand what I'm saying.
Oh, definitely. I agree.
And I like to find metaphors or to be a bit funny. I think it was Richard Feynman, maybe, a famous physicist, who said, if you really understand something, you're able to explain it to the 5-year-old. And this blew my mind, because science is not that hard. And of course, now because of pandemic, more people are anyway interested in science, and I think this is a good time for us, communicators, to get out and reach out and explain stuff. I think anyone is able to understand our basic ideas of what we are doing. They don't need to know the details. It's good if they understand what medicine and science look like nowadays. So I think this is maybe causing a fear from unknown. And by bringing the science closer to them, making it more available, understandable, we are breaking, first ice with them. We are breaking the fear, and we are becoming like normal human beings. Because myself, I thought a scientist is someone who is like always closed somewhere and wants to take advantage of everyone and rule the world. And the truth is, we are not like this. We are just normal people. What really made me realize science is amazing is the approach of all the professors from Nottingham University, my first uni, to lecturing. I was mind blown how passionate those people are. Really, like, I've never seen someone who would explain like very basic genetics, like, boring genetics. They do it in such a way that you just want to stand up and be like, oh my God, like this is all I want to do for the rest of my life. And they really, maybe a bit falsely, but showed me like how amazing the science is without showing me the dark side. But it helped me to realize I'm in the right place. And I wish everyone to basically experience those kind of lectures in their lives, because I felt like everything is interesting because they showed it in interesting ways to us. This was like this whole passion, this big like love to explain someone, because I like writing and talking, and especially when I can feel like I'm being able to explain it in a way that someone is trusting you, that this is really true what you're saying. This is perfect.
But this is actually not an easy thing to do, which is why I've been really impressed with your work. The thing that I find particularly difficult nowadays is that people believe Facebook more than they believe, let's call it, experts. And it can come from the fact that the experts are not using the right language. But it also comes from the fact that people are just more keen on trusting some weird source. To me, it gets very discouraging sometimes. Like, of course, when I talk to my family and friends, and especially now at times of pandemics, we've had countless discussions about several things. It gets frustrating at times because you try to get very basic and explain things, but you somehow lose credibility, especially with your close ones. Like, somehow they don't see you as this expert. Also, it's very hard to put it in the right way and to really get the message across. And I think, we as scientists should be doing this, but I think you also have this like skills that make it a bit easier. You not only do the blog, but you also are writing for the Forbes, and now you had the ebooks, which I also would like to touch upon, even though they are in Polish. I was very impressed that you managed to do it during your PhD, to write 2 ebooks that are... One of them is about cancer. The other one is about COVID pandemics. So this is the more recent one. What I find particularly interesting about this is that it's written in a way that makes it easy for really anybody to understand how the cancer develops or how this mechanism functions. It's a fantastic thing. I guess it must have been very difficult to conceive it in this way. Also, I feel like the illustrations were helpful. I've had several people in my close proximity who had become sick with cancer, and it's always a problem to even understand what their doctors are saying. And it's not the fault of the doctors. I mean, it partially is in a way that they all use all of these words, and, they talk about markers, and no one knows what it means. And I think this kind of resources are super important. I'm very impressed that you managed to get this to work, and also to get this to be so not scary, because that's another thing. You take the topics that are inducing a lot of fear in people. It's very difficult to write it in a way that will not make people more fearful. I was wondering how your process looked like. Was it something that you had in your mind already for some time, especially from your discussions with other people, or was it something that was kind of evolving slowly into what happened in the end. How did this work? Because to me, this is very, very impressive. And this is something that I'm very, curious about.
First of all, Kasia, thank you so much, because hearing it from a developmental biologist who really knows what cancer is, it's amazing. So I'm feeling super grateful. Yes, maybe let's start from, I always try to explain difficult things to myself first. And if I'm 100% sure I understand it, and maybe that's why also my learning and studying takes so long. And when I understand it, then I always try to find a system to explain it to someone else. And this is always happening under the shower, I promise. So I would have this idea and thinking like, how can I explain that, there is not a single drug for a cancer, because cancer is a whole lot of like 200 different diseases, right? And then I'm leaving this topic, but after 5 days under the shower, I realized, okay, let's make this metaphor with an umbrella of having it as another word that everyone understands. Okay, infection. Everyone knows that infection is actually a word that explains any type of infection, from like parasites, bacteria, viruses, and it can happen anywhere in your body. Yeah. It can make you feel like you have a flu, or it can make your skin go weird. I actually try always to find some kind of comparisons from situations that more people understand.
Yes.
And I think Neil deGrasse Tyson does it very well. And by reading people who were in my life idols in science, so like Neil or Richard Dawkins and his selfish gene, or even James Watson, he also wrote about genetics in a way that I understood it being still in high school. I kind of started thinking in the same way. How can I explain it so anyone can understand? And the ebook was meant to be a guidebook for patients and also for their families, right?
Yes.
And there is nothing about grading or about severity of a disease, but there are very basic information that without those, you're not even able to understand what doctor says.
Exactly. And then you know what you want to ask about, which I find very helpful in this book.
And I love cancer. I mean, it sounds awful, but I really like genetics of cancer. And because my passion to cancer was so strong, I realized I need to write something. And also I went for my own, issues, because I had a breast tumor that was removed, and it was like pretty huge one. So it stopped me from working for a while. And then I realized, oh my God, what happens in my head is nothing in comparison to what happens to the girl who has no idea about breast cancer and is just like flooded with this information online. And I found a publisher who was very happy to do it in the way I really wanted. So I really tried to give people the very basic, and merging this with real life situations and with real life things that you can do, basically. So it's not like we're all going to die of cancer. There are things that we can do to minimize the risk.
Mm-hmm. Yeah, I think it's super important resource. For Polish listeners, I really recommend you have a look at the book. As you mentioned, it's not only good for people who get sick, but also for their families, to understand it better, what is happening, to understand how it works and how it appears.
Yes, it's a sad thing, but also it happens all around us, that we're really supposed to be equipped with this knowledge. We live in a world that, every other of us will get cancer at some point, so maybe it's better to know about this before and know how to prevent it. And this is the same thing what happened when I was writing COVID ebook. First of all, for a year and a half, I was really just kind of showing my knowledge and researching all the news coming from COVID, looking for really like the most recent papers and showing to people in like an easy to digest way.
Yes.
And I wanted to close everything that I touched upon in a one book of some sort. And the same publisher just like agreed to do this again with me. We published this ebook on COVID, first of all, because the questions that people were asking me, I know they're coming from their not understanding the basics of immunology.
How the vaccines work, how the virus reproduces, all of these things.
Exactly.
50% of people out there don't even understand what's the difference between bacteria and the virus. It's a very important thing to actually understand why we need certain ways of protecting ourselves because we cannot use antibiotics for COVID.
People don't know, but they still form opinions. And this is the dangerous part, because you're supposed to base your opinions on some kind of fact, some kind of knowledge. If you don't have this knowledge, your opinion is just someone else's opinion repeated. It's okay if it happens that you get your opinion from someone who is actually knowledgeable. But if you get it from someone who is just showing some kind of emotional post on Facebook, this is not good, because you can self-harm by not vaccinating yourself.
Definitely.
Because you didn't have enough knowledge. It's basically a guide of how to understand pandemic, and also a book that you can use for any future pandemics with viruses included.
Which are going to happen. Yeah, maybe not to the scale, but I mean, we've had several in the last years. It's not only COVID that happened.
The small outbreaks will happen, yeah, for sure.
Exactly. It's gonna go as also the density of population increases. So now I want to go to the more classical questions. You are working really hard, both working in your lab as well as doing all of the freelance stuff, the Instagram, the popular science, the psychom. I wonder, how do you still keep your work-life balance in check? So what do you do to still have some time for yourself, to still have some time for your close ones, for your fiancé, and to not get all worked up in this? Because I must say, I'm just juggling PhD and the podcast, and sometimes it feels like it's a lot, and it's just these two things. So yeah.
It comes from the times when I used to be a question writer. I think this allowed me to train a lot of discipline in myself. And also, different people pursue resting or relaxing in different ways. I cannot really say that some of my activities that I'm doing, I perceive as work and something that I need to rest afterwards. For example, what I'm doing on Instagram, I know some people treat it as work, but for me, it's free time. I have the system of working that I spend 45 minutes on doing something and then I change because my brain gets tired and I'm not so productive anymore. So I'm doing my Instagramming, for example, during this 5-10 minute break between one task and another. With time, I just became quite efficient with doing even a couple of sentences of one post and then moving on to the next thing and coming back to it when I have time. Of course, there are times that I was super tired just all the time. And for me, it was a great achievement if I managed to do just basic stuff together with work. And sometimes I'm also working in the lab from 9 till midnight and I don't have time for it, right? But I think I always try to have a bit discipline in me.
Mm-hmm.
And also the way how I'm spending my free time, I try to do it actively, let's say. So, for example, I live long distance with my fiancé. So when I have a weekend that I'm not spending in the lab, I would drive there. It's 6 hours. So during the 6 hours, I would listen to podcasts to do stuff from my to-do list. And still, this is a fun thing because you can listen to a podcast. It's nothing super tough, right? Driving is not so tough on the German motorway. And then I arrive there and I always try to have enough sleep. This is my holy grail. I always try to sleep enough. Some seasons, maybe within a year, I can do with 6 hours of sleep, but some it's 8. And I think because I sleep well, then I'm basically more active during the day. Yeah, and I don't eat so many sweets because when I eat sweets, I actually can see that I'm less productive. So really, I'm trying to look after myself in a way that I tell other people to look after.
That's good, yeah.
And when I'm feeling tired and I know, okay, this is the time when I'm supposed to keep my balance and rest, I would go for a walk or a run or a jog, depending on how well I feel. And it's still resting because my brain rests. But realizing it in your life, the earlier you do, the better probably. What you have to do is really set up your priorities, right? Because when you have so many tasks, you have to really choose the ones that are the most important. And those you will do during the day and the rest when you have time or not. Really listen to your body. My life-work balance, maybe it's not perfect for some, but I think I'm feeling the best when I'm actually actively seeing my friends, visiting my family, spending time with my horses. I do a lot of stuff that I don't share on Instagram, maybe. People don't know that, I'm crazy about my horses so much. But this is time when I have my phone in my pocket, hidden.
Yeah, this is good
and I don't use
unplug and just enjoy it.
Exactly. I think people just tend to waste time a lot. And this is something I cannot afford because it's a sinusoid, right? So when you feel good and you're on the top of the things, you can do everything you want. Yeah. But there will be times, there will be months when you feel worse. And then you just do half of your to-do list. And it's still fine because it's anyway a big to-do list, right? So I let myself rest whenever I need. But I try to use the energetic times, in the best way possible.
I was like this for most of my life. I would just really do a lot. I would be in the lab, but then I would do some online courses at home. I had always something happening. What I find particularly challenging for me now that I'm sick is that my body, it's like sabotaging me all the way. And this is very difficult for me to adjust because I was doing so much and now suddenly not only I have to do less, which is something that you can adapt to, but it's just like I have sometimes no idea which day is going to be a good and which day is going to be a bad day. And it's really frustrating. I feel like I will adjust to this. But hearing you, I can see myself in this, being very dynamic and being very organized. And currently I'm going through this phase of I really don't manage to do all the things that I want to do.
Yeah, the most important is to adjust and adapt to what your body is telling you.
Yeah.
Because, when I was recovering from the operation, of course I couldn't move much, but I was feeling, okay, I cannot lie in bed because it makes me just depressed. And then I was asking my boyfriend back then to just go, cross-country skiing with me. And of course I didn't run, but I would just super slowly go around mountains and just look around and just get my energy out of nature. And those little activities, you remind your brain that you don't have to go for a run if you don't have energy for it. You can go for a walk. I'm starting to treat my body as my best friend. And in the past, I treated my body as something I can use, like a tool.
I'm still in a frenemy kind of phase, I mean, I know that I will get there. It's just, it's a big adjustment.
This body helps you to achieve so much, and now your body needs to rest.
It's completely new. And then you just don't know yet how to sometimes read what happens. I feel like I'm currently in this stage. Okay, I have 3 last questions. I always like to ask about room for improvement in science. Yeah, I was quite surprised how broad my guest answers are. But on the other hand, every one of us has a thing that is particularly important to us that we find particularly, timely or just personally touching. So what would you think for you is something that needs to be improved, needs to be changed, either in science in general or, in academic world or in PhD world? What would you say is something that you find particularly in need of improvement?
I don't even know where to start. That would improve everything. First of all, I think university labs should be more like companies, because it bugs me so much that we spend hours in pipetting and doing things manually, and then in the company they have a robot. Of course, it's known that in academia it's never going to be so fast and so efficient, but I think we should take more from the companies that we do, actually. Even looking at the PIs, if they become more like bosses in companies, who are first of all accountable, who need to be still trained into how to be a good leader and a boss, and they need to take care of their workers because they are checked if they do things properly, and they're also rated by their workers. It will be completely different, because we wouldn't have the situations that some people are out there because they have a big surname, but actually they're very nasty people. So this, and also some kind of initiative towards PhDs like companies take towards their workers, for example, space to just rest during the day. And also in some universities, it already happens, but I think not in the one I'm at the moment, that people are collaborating a lot with themselves. Labs which are in one department. Seriously, we don't need so many equipment per each lab. We could share some things, right? I think science would end up somewhere else if people were just talking to each other, not competing with each other, but actually
definitely.
talking. And there should be well-known papers with negative results. Seriously, results that never worked, drugs that were checked.
So that we don't waste time repeating these things.
Yeah, the clinical trial didn't work. We put it in this paper, and it's still a publication because we tried, we spent time on it, because we make the same mistakes over and over again, because nobody managed to write anywhere that this doesn't work,
Yes. I mean, it's starting, but
it's never going to be, it should be compulsory.
It would save money, it would save time. In a big scheme of things, moving science forward, this could help save time.
Yeah, we wouldn't be so blind anymore.
Yeah. No, I think this is important. Like everything that you said, some of these things were brought up before, especially the collaborative nature of research. We should go away from our own egos and, thinking that we have to be the best as a unit, and just thinking a bit more about how much more you could do if you didn't have to, let's say, learn all of the technical skills, but you could ask someone who is much better than you, can do it faster, and just collaborate and move forward. I definitely agree. I mean, accountability of PIs. This hasn't been brought up here, but this is something that when I talk to my friends and a lot of different PhD students, it gets brought up a lot. I mean, we had recently this example of a very prominent PI being, released from his position. But I mean, we start having these things. Like I've heard that in US, they were revoking grants for people who have been found to have some sort of mobbing issues with their workers. And stepping out even from this, I feel like at universities there is lack of structures that would provide enough support for people to come forward when there is such a situation that needs help. There is not enough structures to, first of all, to just feel safe to come forward.
Yeah, we don't have anywhere to go.
Yeah, to also feel like this is okay to say that something is wrong, where you feel like I'm just a PhD student. What can I do? And this is this big person, and when you feel like there is no support, it's a very difficult fight or like struggle that you kind of have in you to do this.
Yeah, the whole system would have to be changed because, in a different uni, not in my uni, my friend has now issued that after 3 years, she needs to change her PhD lab because she knows some things are just very not right in the lab, and then there is no system for her to escape. So I think it's very complicated, and there are some countries and some universities that have very good structure, like Nottingham. I cannot imagine a PI in the UK asking me, when I want to get pregnant or things like this. And here, I think it's just an everyday life, and then people live with it. And I'm like, well, this is not correct. It should be changed. But who wants to risk their position to start changing something?
Exactly.
And how high can you get?
I mean, it's a systemic issue, right? Okay, moving forward to the last part. It's the part that I called cool science. One thing is, if you had unlimited amount of money and you could just study whatever you want, or you could have the technology that you want, what is something that you find particularly interesting and something that you would like to try to do?
Yes, I think I would go for deep cancer genetics. I would like to research exact changes that happen during the development of any If we are going so far with unlimited amount of money, then I would say any cancer, and then maybe reverting it back, right? So some kind of genetic tool and knowledge to be able to revert or stop the processes coming. Because we know that cancer is basically this one mutation too many in the cell, and to stop this, at least this one last mutation would be already amazing. So for sure something deep in cancer. And I don't think I would ever go so far away from research topics that I'm already covering, because I just find it so fascinating, right? So something like alongside medicine, cancer and genetics, it's just so unknown still to us that it would be nice to finally find out more things.
Yeah, it sounds really cool. You would need a lot of money, so definitely. Okay, so last question is the question about the dinner. I don't know if you've heard it on the podcast, but if you could have dinner or coffee with someone that was an inspiration to you, or you find particularly interesting and you would just like to chat, it can be someone that is still living or no longer living, who would it be?
A scientific part of me really wants to see Marie Curie, because she's been my biggest life inspiration in terms of like female scientists. My second name is Maria because of Maria. My mom studied chemistry as well, but in a technical school, and like she was also amazed by Marie Curie, and so she named me after her. And I just would like to talk to someone who managed to get so many Nobel Prizes and, has so many ideas. And, what amazes me, she had a lab next to her house. It's nothing, a big institute and stuff. And non-scientific parts of me would really, really love to have a coffee with my already dead grandfather, because I feel like I lost him too early. Mm-hmm. And there was still so much to discover. And I think the older I am, the more I feel like I was just too young to understand him. And he was basically the person who taught me how to read, what to read. Mm-hmm. He used to read newspapers and then highlight the stuff that should be interesting to me and pass it on to me. And he always had this like very, very intelligent conversations. And when I didn't understand, he used to be more like a humanist person. So psychology, history, geography was his main field. When I didn't understand, grandfather, who Churchill is, he used to draw me the story of Churchill on the
so cool
on the desk and show it to me. And I think I was just too young to realize how much of a knowledge source and life experience he is holding on to. Before he can share it all with me. And I didn't appreciate it at times, I must say. I was just too young. And I was always he is very talkative and come on. And now I think, wow. Yeah. And, I'm actually renovating a house where he used to live. And we're going to move in.
Oh, soon. That's nice.
And to me, it's such a turnover, life makes circles all the time. His death made me drop horse riding and start thinking about university studies for real. And this is why I left to England. This was a huge event in my life. And I think now I would just I would love to talk to him and be this, person who can also bring something from me to him.
Yeah, I'm sure he would love to have this kind of conversation with you, I can understand this. I think partially me, I've lost several people when I was young. Also very close. I lost my mom when I was just 12. And, just to imagine, when you're 12, what would you, talk to your mom about? Some stuff. But there's so many things that I would like to actually bring up right now and just see what she would think about. Yeah, I think it would be cool for you. And he would definitely be proud that you went this path.
As much as your mom about you.
Yeah.
Yeah, maybe those stories, shape us in the way we are. But actually, out of all those amazing, bright minds I could talk to, I think those close people are just the most...
of course.
The most precious.
Thank you for your honesty. And thank you for sharing so much about your science. I mean, it's clear that you are passionate not only about your own work, but just science in general. And I'm also very appreciative of all of the work that you do to make science more accessible to general public and to people. And I'm very glad that, we can share a bit about you with other people out there. Thanks a lot.
Oh my God, I'm gonna cry. Yes, thank you very much for inviting me, and I hope scientists and non-scientists will enjoy this podcast. I know at times it was hard to maybe understand for people who are not sciencey, but our work, both of us, is also to support other scientists. So I hope maybe it will give a lot of motivation and inspiration to future generations.
I'm sure. Thank you so much.
Thank you.